I'm joined today by Gareth Griffiths, the scientific advisor at Inaphaea. Gareth, give us an introduction to yourself.
So, just to introduce myself, my background is very oncology focused. I did a PhD in cancer research, and this was in an immunology department. So, I have good knowledge of immunology and cancer as well. I then moved and did some postdocs at the University of Manchester. I worked at the Patterson Institute at the Christie Hospital in Manchester, and I then moved on to a job role at AstraZeneca, where I was developing assays for high content screening. After that, I span out a company from AstraZeneca with a colleague, and this was called Imogen Therapeutics. And over the next 10 years, we acquired the biobank of patient-derived cells that Inaphaea has today. And this was a company that grew to 60 people at the time before the assets were bought by Inaphaea.
So, Gareth, for those who haven't heard of the company, can you give us an introduction to Inaphaea?
So Inaphaea is a pharmaceutical translational service company. The aim is to improve translation of drug candidates into the clinic, specialize in oncology, and it's a company that's based at Medicity, Nottingham.
Gareth, can you talk about Inaphaea's labs and the key equipment that your clients benefit from?
Yes, Inaphaea Labs are a BSL-2 facility, and they can carry out standard tissue culture. Now, this could be 2D or 3D. The capabilities are 96 and 384 well formats for drug screening. Um, we have automated drug dosing, so this is a very rapid kind of dosing, no manual input is required. We also have live cell imaging capabilities. Um, Particularly, we have high content imaging platforms. We have three of these where we can do higher resolution imaging and phenotypic assays. We're also capable of running cells normoxic and under hypoxia as well to give a more clinical sort of field setting to them. Um, and we also have the gold standard Xcelligence platform. This is a screening platform that is particularly well used in immuno-cold culture applications. We can measure impedance on the plates, as well as imaging and live imaging over 3 days, 5 days, 7 days if necessary. We can also do label-free morphological analysis, and we have over 100 fixed endpoint assays, as well as custom assay development available in the lab. So we have a whole range of different equipment and capabilities.
So, Gareth, please can you tell us what a patient-derived cell is?
A patient-derived cell, or PDC as it's abbreviated to, refers to a cell that is isolated directly from a patient's tissue sample. In our case, the cells are isolated from tumor samples. These cells are only cultured for a short term in time in the lab, therefore retain genetic and phenotypic characteristics of the original tumor, making them a powerful tool for personalized and translational research.
So, how does a PDC differ from a standard cell line?
Well, patient-derived cells provide a closer approximation to the original disease state compared to traditional cell lines. And this allows translatable drug screening, the understanding of resistance mechanisms that arise in tumors. Typically, a cell line is cancer cells grown indefinitely in vitro, originally derived from patients but passaged extensively over time. Some of the commonly used cell lines were derived in the 1950s and 60s. For example, the U87 cell line, which has been extensively studied since 1966, was recently shown to have no resemblance to the original isolated tumor cells. And this calls into question how translatable the data is that has been generated from these models in a clinical setting. With low passage PDCs, they are, of course, more likely to be clinically relevant because there are only several passages.
So, why would you choose a patient-derived cell over a cell line?
Traditional cell lines are a simplified model, and whereas PDCs retain more characteristics of the original tumour and are less altered by the extensive culturing.
Where does a patient-derived cell fit in terms of cell lines, organoids, and xenografts?
Okay, so cell lines are traditionally used by most companies and academics that go for simplicity, higher throughput screening, ease of use, but they lack all the clinical data behind them and the characteristics of the original tumour. PDCs, these are tumours that have been acquired recently from patients. They retain more characteristics of the original tumour, and they're less altered by extensive culturing because of the low passages that we use them at. And the Inaphaea biobank, I should say, contains approximately 470 PDCs across 66 different indications, all with these low passages, and therefore they're an ideal tool for early stage drug discovery projects. Organoids, on the other hand, are 3D cultures created from patient-derived cells mimicking the architecture and functionality of the original tissue. They're heterogeneous and do not consist solely of cancer cells, and thus more closely resemble histological features of the original tumour. PDCs can serve as a source for PDOs, allowing researchers to create a more realistic tumour model. PDOs provide a 3D environment that captures more of the tumour's microenvironment compared to traditional PDCs cultured as 2D cells. Patient-derived xenografts, on the other hand, or PDX models, involve implanting patient-derived cells or tumour fragments directly into an animal model. PDXs maintain the tumour's microenvironment and native cellular heterogeneity, closely resembling the patient's cancer in its growth and response to treatments. These models are excellent for studying tumour growth and metastasis in vivo, testing drug efficacy in a living system.
Gareth, can you introduce the RNA-seq part of Inaphaea's offering and explain the importance of target selection?
So RNA-seq or RNA sequencing can play a crucial role in selecting the most appropriate cancer in vitro models to test a particular drug target. In the Inaphaea biobank, there are over 300 models with RNA-seq associated data. In target validation studies, RNA-seq can determine if your drug target is expressed in a given PDC. By analyzing RNA-seq data sets, you can therefore identify which PDCs have high expression levels of your target gene. This ensures that you're selecting models where the target is active, making them suitable for testing. For example, if your drug aims to inhibit a specific receptor overexpressed in breast cancer, RNA-seq can indicate which PDCs show high expression of this target, and that allows selection of the PDCs in your study. RNA-seq data also allows development of machine learning AI models, revealing hidden mechanisms of resistance and being able to predict response to specific drugs, thereby improving the chances of successful preclinical validation.
Can you define why a patient-derived cell fits in with Inaphaea's approach to bringing clinically relevant data earlier?
For patient-derived cells, because they provide a closer approximation to the original disease state compared to traditional cell lines, it makes them a powerful tool for personalized and translational research. And this is because they preserve the genetic and histological features of the original tumor, and thereby it's a realistic model for studying the different cancer biologies and drug responses in vitro. PDCs can be directly used for 2D in vitro studies and rapidly screen oncology drugs. This means that drugs can be progressed or eliminated at this early stage. Drugs that progress can then be screened in a more relevant 3D organoid type model. At Inaphaea, we have not only isolated the cancer cells, but also the tumor-associated non-cancerous stromal cells for each sample, such as cancer-associated fibroblasts. This allows us to recombine these as a 3D model and perform rapid screening in standardized organoid models. Following on from these in vitro studies, Inaphaea can progress drugs in vivo models. Inaphaea are working with collaborators who can take our PDCs and implant them into in vivo models such as mice or zebrafish, allowing us to measure such endpoints as tumor growth and metastasis. So in summary, Inaphaea can lead you through the drug discovery process from model selection using RNA-seq data, 2D screening assays, 3D organoid models, and finally testing in an in vivo model. In essence, PDCs are highly relevant models for drug screening and play a pivotal role in drug development and personalized cancer therapies.
So Gareth, can you introduce Inaphaea Biobank?
Yes, the Inaphaea Biobank is a specialized repository that collects, stores, and manages patient-derived cell models and associated clinical data for research purposes. It's a living biobank of around 500 patient-derived tumors which have been taken from cancer patients over several years. These PDCs are essential for cancer research and drug discovery, offering access to a wide variety of cancer models that represent different types and subtypes of tumors. Currently, there are 66 different indications represented in the biobank. With over half of these models having the associated RNA-seq data, it's possible to interrogate the bank for specific target expression and tumor type, making project design a straightforward process.
Introduce us to the Inaphaea PDC offering of services and products.
So Inaphaea can offer a range of preclinical services using its PDCs. Cancer cell assays. These are designed to evaluate drug efficacy using various PDCs. They measure parameters like cytotoxicity, cell metabolism, and apoptosis markers to screen drug candidates' activity and mechanisms. High-content screening is a key part of Inaphaea services. Possessing 3 different platforms for image-based assays, we can run both fixed endpoint and kinetic cell-based assays. The lab is also kitted out with a range of automated platforms, such as automated drug dosing, and this allows us to carry out a rapid 2D PDC drug screening service. We also have our 3D organoid screening service, a closer approximation to the patient tumor, which ensures more clinically relevant data. We realize every customer is different, and therefore custom assay development and project design is something we are highly experienced in. We can offer advice and help in many of these areas.
So can you describe the products you have available at the biobank?
We have a large biobank of many different PDCs, and we understand that some customers are keen to carry out some experiments in their labs. We therefore provide an option to buy assay-ready vials. Inaphaea can ship selected vials for use directly to the client's lab or another CRO. The purified cancer cells, stromal cells, or mixed populations are also available for purchase in certain models. Licensing of PDCs, annual license, or outright purchase is optional. If a PDC isn't available in our biobank, we can source the material through our network of clinical providers and develop your PDC of choice.
Now, most people have heard of the importance of immuno-oncology. Can you tell us what services Inaphaea can offer in this area?
So this is a big area and one that Inaphaea is striving to provide high-quality, relevant assays for. We understand that approximately 40 to 50% of companies are focusing on immuno-oncology therapies as a core part of their drug development pipelines. It's a rapidly growing field which has shown significant promise in harnessing the body's immune system to target and destroy cancer cells. Unfortunately, the majority of patients do not respond to immunotherapy, making the identification of predictive and mechanisms of resistance an area of intense research. At Inaphaea, we are able to utilize our RNA-seq data in order to identify cells with targets of interest, such as immune checkpoint markers such as PD-L1. We're able to use patient blood samples from donors, collect PBMCs, and isolate particular cell types for use in immune oncology assays. For example, a T-cell killing assay to measure the ability of T cells to recognize tumor cells and kill them. Inaphaea also has an Xcelligent system in-house, which allows kinetic-based T-cell killing assays to be carried out over several days. The immune system is complex, and multiple other cell types can play a role in cancer cell killing. Macrophages, neutrophils can also kill cells given the right conditions. At Inaphaea, we are capable of carrying out a whole range of these assays using cold cultures of immune cells.
So what are Inaphaea's future plans and ambitions for its biobank, products and services?
There are many plans for Inaphaea in the near and long-term future. The biobank is a large resource, but we intend to keep adding to the bank through new tissue acquisition, We also intend to diversify the biobank and focus on some of the rarer cancer types. We also intend to take the same patient blood along with the tumour. This is important as currently donor blood is used, and this can be inconsistent and not truly representative. Having autologous matching immune cells clearly represents a far more accurate model with which to test drugs. So in essence, we are building the biobank, increasing that diversity in order to offer highly sought after models and expanding our lab services, especially in the immuno-oncology area.
Gareth, thank you. It's been great to speak to you today and hear about Inaphaea.